Okay, good afternoon. I think without further ado, we'll get the workshop or the educational session on computational neuroscience kicked off. Stefan and I have done this course now for about 8 years. I think we had a break last year, and we've completely re-engineered the talks. Just about every speaker has a completely new talk. I think in the past it's fair to say we had quite a lot of dense maths, which was probably not at the appropriately entry level. And so we have gone for a more conceptual approach and really would value your constructive feedback because we really like holding this workshop. Computational neuroscience is a really quickly growing field, and basically if we make it too dense, then we get bad votes and it will disappear from the program. So I say that quite openly. Moving on to the session as a whole, you'll hear a really broad repertoire of talks, computational models of the human brain, right through to models of behaviour, inference, decision-making, and then a lovely open source virtual simulation program. And we have some fantastic speakers. Our other co-organiser, Jean Denazou, on the other hand, is sitting psychology exams and is not here. So I'm going to give an introduction to computational neuroscience. It's biased towards what I do, which is biophysical models of the brain. And Peter will be after me, Peter Robinson, and he also has that approach. I do have a few slides that move further towards behaviour and inference, but we'll leave those topics more to the experts later on. So, one thing that intrigued me, having studied applied mathematics and physics at university, were these grand theories of how things worked. So, we had theories of the atom, of the universe, and of electricity. And each of these were captured over sometimes hundreds of years by leading thinkers in the field. But what would we say is the model of the brain? Some would say the best model of the brain is a brain, and possibly even the same brain. And I'm kind of half joking there, but it's not completely off the mark with some of our colleagues' approach. Some would say that we need to understand the neuron, ion channels, membranes. These are the computational units. Of course, the human connectome is a wonderful model of the brain, or certainly an important piece of the brain. But what those of us that you'll hear quite a lot today think is that the models of the brain should be like models of other systems and therefore based on mathematical descriptions of the system of interest. This is about the only equation I have in my talk. It's dx to t equals f of x plus an input. And I'll just describe that in a little bit more detail. And then we don't observe these states, we observe other measurements of the brain, and I'll speak about that. Most of the equations that you'll hear to some degree involve two components, local circuit processes, which I've caricatured like this, and inputs from other parts of the brain through the connectome, or through local cortical connections. Now, when we're talking about a model of the brain, a model that we measure at the large scale, we're not going to be talking about models of individual spiking neurons, because these are good models for that scale of description, but not the large scale that we typically interrogate in the Organisation for Human Brain Mapping. And what people in the field have done, and Peter's one of the pioneers here, is take a local average, mean field averages, over thousands or tens of thousands of the individual elements. That's like the mean field approach to the temperature in this room. We wouldn't try to describe the individual motion of the atoms, but just little elements. And if the room was more or less homogeneous, we'd just have one temperature. Okay, how do we go about doing this? Well, we're not going to model the brain as the brain. We're going to use some level of abstraction. So pretty much all the talks that you'll hear today embody some level of abstraction. and pretty often some type of simulation. And I'll come back to these fancy looking waves in the brain. I'm stealing a bit of thunder for my talk on Tuesday here, but nonetheless, I think they're instructive. Now, these levels of abstraction can still leave us with very detailed neurophysiological or neural field models. These contain all sorts of things about neuronal states, firing rates, dendritic filtering. I think Peter will touch on these. The thalamus, cortical delays. But not every nut can be cut with, opened with that hammer. So sometimes we have more abstract models. And I've put here the classic equation used for DCM, but we have other algebraic equations, including something called the Kuramoto model. And one can move between the two. One can take these models and do something called a linearization and a Taylor series truncation to get these. And then one can hopefully move between the two levels of description. Now, the other important thing, as I said before, is that we don't measure the states. We have some forward model that maps the neuronal states into some sort of observables. And this is some work that Peter and I worked on with Kevin Aquino. And we have a very detailed forward model from neuronal states to hemodynamics. And that gives us a prediction of the bold. And then we can take a brain and we can do a measurement, we can get an observation, and we can look at the match between the prediction and the observation. That would be for fMRI. If we wanted to predict EEG data, we use a different observation equation. This is a forward model. And if you've got a particular type of model, particularly the more simple models, you can take an invariational scheme, a Bayes-based scheme, and you can actually invert the model from the data and recover some estimates of the model's parameters. Now, this is why we might use the more simple models, because the more complex models are more difficult, particularly the nonlinear models, but there's a lot of work in that area. And you'll hear some of this in the talk this afternoon by Rosalind. Now, what I've done in my career is something called neural mass models. So we take a little caricature of the cortical column. Most of my work is just based on one population of pyramidal cells and some local inhibitory cells. And the model that I'll show here, the pyramidal cells are conductance-based and they have a fast time scale, and the inhibitory cells see the spikes, they begin to fire very slowly, and then they damp the spikes. And if you put that into MATLAB, you can always email me for the code. Hopefully there'll be a toolbox pretty soon. In fact, there is in the virtual brain. These are the fast time scales, the pyramidal cells and their potassium channels. You see this slower time scale of the inhibitory cells, turning the pyramidal cells on and off, and you get this bursting. These are the time series. And if we take T out and we plot the three against each other, then we get something called a phase portrait, and we get an attractor. And this beautiful work was pioneered by a French mathematician, Henri Poincaré, about 130 years ago, and it's a geometric description, and that's what I look at a lot. So you can see the bursting is captured in this particular beast. If we change one of the parameters, we go through something called a bifurcation, and we get, in this case, chaotic dynamics. And so we see that there's sort of incongruent timescale mixing, and we get this lovely looking strange attractor. Who's heard of chaos theory and strange attractors, by the way? So pretty much we're charging into neuroscience with these sorts of models, but we still have a way to go. Okay, so that's good and well and fine for my local circuit equation, and I promised I was going to couple these up through the connectome. So here they are, caricatured like this. Here's a picture of the connectome from a paper by Alistair Perry, just published in Neuroimaging. And then we make the coupling between them according to these structural connectivity matrices, and we fire it off, and off we go. And this work we've published, Chris Honey and I, and Olaf Sporns and Rolf Cotter. Gustavo Deco has published a lot on this, and you've probably seen a bit of that work, and Gustavo often presents in this workshop. Peter's also done quite a lot of work here, and he'll speak of this. Now, in these funny-looking movies, the states are now represented in a colour. And I've taken the chaotic oscillator, although a lot of the chaos has been suppressed, and that's hopefully you can see the nodes each have a colour according to their states. So if we do that, what do we get? Basically a whole lot of beautiful looking self-organised patterns. They come in a variety of forms: rotating waves, breathers, travelling waves. These are all exactly the same set of equations, and these organise themselves at the global scale into these particular dynamical patterns. So do they just get stuck? Does the virtual simulated brain get stuck in these? So if we watch them for long enough, you'll see that they typically start with one pattern. And I'll have to pause here. Hope I'm not running out of too much time, Stefan. I'm about halfway through. And after a little while now we see that this has gone from doing this. Now there's this other patch here that's appeared. So now we've got two waves in each hemisphere. And after a little bit of time then that becomes unstable. And we transition to other waves. Sometimes it slows down just before a transition. Okay, and then it settles on another pattern. So James Roberts, who's a physicist in my group, did his PhD with Peter, came up with a measure for this based on taking the Hilbert transform of all the nodes and looking at the left and right hemispheres. Did you see? This is the cross-correlation between the left and the right hemisphere with time delays, so this is zero lag. So you see that according to different patterns, have different patterns of synchrony between the two hemispheres. And between the patterns, it goes through a period of low chaotic order, and you see the periods, the little light blue stripes. So we can use those and just very automatically partition the time series into these different patterns. These are called, it's a very complex form of it, but it's called a heteroclinic cycle. So it's a system which organises into a particular pattern, looks hunky-dory, but then the pattern becomes unstable, it loses order, goes into a different pattern. And this will continue indefinitely. It's also called metastability, and you've probably seen a little bit of that in the literature. But we know these are metastable states because we've looked at the equations and we've looked at the states. Just a word of concern is that these are properties of the model. They're not properties of the data. And so I caution a little bit measuring fMRI and saying it's metastable. You really need metastability as a complex property of a particular model. Okay. So here we are, all satisfied, and what I said before is that we should take in our framework these states, push them through a hemodynamic forward model. That says if there's neural activity, then there's a glial signal, then there's local vasodilation, and then there must be a system that obeys the laws of physics. We get changes in the hemodynamics and blood oxygenation, and then we can get an observable. So we can predict fMRI data. And actually, if you do that, well, I've actually shown some fMRI data. Andrew Zaleski and Alex Finito and I and some others uploaded some of the Human Connectome Project data. You've probably seen this approach where we window the data into small windows and look at the cross-correlation matrix. And you get these, in this particular paper, which was published last year, transitions between highly ordered states and states with low order. So this is what you'd say, I said at the beginning, where you're predicting something from your model. Now, if we measure this, we can't really say it's metastability unless we try and invert that. So I hope I've made that point clear. Now that relies upon breaking the brain into little parcels and having one state or two or three states at each parcel. You don't need to do that. Sometimes you might say the scale that I'm observing the brain, we don't really see these little patches at about 300 microns. So we can treat the brain as a continuum. And we replace this coupling through the connectome with local coupling here and bring in a what's called a damped wave equation, and Peter will speak more about this in his next talk. So this is the Robinson model with the corticothalamic delay and feedback. And we have dendritic filtering of the currents. I'm not going to go into any detail, just to say this is the formal relationship between coupled neural masses, which you'll see people like Petra and Gustavo and I do, and the neural field models of people like Peter and me. Okay, I'm just about finished. I just want to quickly give you some snapshots to convince you that this approach has worked in a number of areas. It's been 5 minutes, good. Quite a lot of work over about 15 years with a lot of collaborators. Frank Freya and Petra and I and Peter looked at the neural field model I just showed and used that as a generative model of multi-stable cortical rhythms. So that's one success. James Roberts and I looked at these very stochastic-looking eye movements. So we're not talking about brain states, but eye movements. There's 2 movies here that weren't working. I'll have to reformat those. But we have another one of these fancy-looking equations for eye movement. They're healthy states. Very recently I worked with a wonderful neurophysiologist, Samsa van Hartelo, and we looked at these patterns of burst suppression in neonates either born following hypoxia or before 30 weeks gestation. It's quite common. And we came up with a more of a phenomenological model for the burst suppression. And I think Peter will also talk about using his neural field model as a generative model of seizures. So at least with electrophysiological data and more recently with fMRI data, this approach appears to be having some success. It's not without controversy. There's still a lot of people internationally who think spikes do the coding, and these are epiphenomenological levels of description, and I think personally that's a very creative and constructive dialogue to have. Final 2 minutes. What about linking brain and behavioural models? So I think the computational approach to human behaviour these days is based on a Bayesian approach, which Stefan will talk about, and it comes from a lot of very compelling work showing that when we integrate information from different senses, in this case the haptic sense and the visual sense, each representation has a value, but there's also in perception some uncertainty associated with that value. Now, if we go to our models, I've said there's only a mean. You can think of that as the value of the world that that particular part of the cortex is encoding. But in this approach, there's no such thing as uncertainty. So one of the things that we've started thinking about is replacing these mean field models with something called the Fokker-Planck. Please do not worry about the form of it. It's just, this is more like this guy, but this comes in with the dispersion. So now the states have a mean and they have a variance. Of course, this was described by Drs. Fokker and Planck. And when I first heard Lee Harrison and Carl Friston talk about the Fokker-Planck in neuroscience, I thought it was kind of a bit crazy, but now I'm kind of convinced that's the way to go. So we do what I showed before. We couple the neural masses with Fokker-Plancks, or fluctuation expansions at least, and then we're somewhere close, if we've got hierarchical connections here, to these hierarchical connections between means or values and their uncertainties or volatility where's Christoph? Oh, the beast hasn't turned up, but he'll be speaking about this, and so will Stefan. Okay, so I'll close there. This is the work, this is my group. We're having a lovely time here in Hawaii. So the work that I showed has been conducted over about 15, 20 years with a lot of collaborators, some of whom are here. And before that by people like Walter Freeman, Paul Nunez. As soon as you start mentioning people, you realise you forget people. Lopes da Silva. There's a lovely history behind this work. And in the 70s and 80s it was done fairly quietly, and now it's becoming a lot more popular. Okay, so hopefully that leaves me with a few minutes for questions, and I hope you have some. Okay. So I'll do a little survey. Who's heard of these neural mass and neural field equations? Okay, quite a few people. I think if we had have asked that question 8 years ago, probably far fewer people. I'm interested who in their heart of hearts thinks that these are purely convenient ways of describing the data versus who thinks that maybe there is something to these states and their uncertainties that might be the right level of description. So who thinks spikes is where it's all at, everything else is phenomenological? Good. Good. At least we have some dissent. Eugene Izikovic, at a meeting I was at in San Diego last week, he's a great thinker, very influential. He's quite adamant that every spike matters. And if you look at the Human Brain Project and a lot of work under the Obama Brain Initiative, you'll really see that the microscopic level of description is still very dominant. But I think we're getting to the point of some degree of parity.